Hello, my name is Joshua Mugabe. I welcome you to Osmosis. The left-hand picture, the plant is wilting from lack of water. We have our picture here. It shows that the plant is wilting. As you can see, the leaves are becoming bent or curled, and that is what we are calling wilting, because it lacks water. Osmosis is a special case of diffusion. Osmosis is a special case of diffusion. Osmosis, this is the movement of water molecules from a region of high concentration to a region of low concentration through a semi-permeable membrane. And diffusion is the movement of materials from a region of high concentration particles to a region of low particles. But the difference between osmosis and diffusion is that for osmosis, there is a semi-permeable membrane. And diffusion, we don't have the permeable membrane or semi-permeable membrane. Osmosis involves the diffusion of water through a membrane. Water is going to move through a membrane, and that diffusion or movement of water molecules from a region of their high concentration to a region of their low concentration through a membrane is what we call osmosis. In the beginning, we talked about this plant, that the plant is wilting because it lacks water. What is happening is that the plant is losing water to the atmosphere because of the diffusion of water through a membrane. And when the plant loses water, then it is going to start curling or bending, or what we call wilting, because it has lost its water by diffusion. The membrane may be artificial or non-living. For example, cellophane. We are talking of a membrane which is semi-permeable. We can form an artificial membrane or a non-living membrane like cellophane, and can be used in the experiment to show us the process of osmosis. The membrane must allow water molecules to diffuse through. It is permeable to water. So when we look at our diagram here, I want us to study it very well so that we can understand. We have a membrane in the middle here. You can see that membrane in the middle, and it is separating water this side and this side. This water this side is a dilute solution, and this water this side is a concentrated solution separated by a membrane, and this membrane is permeable to water, meaning that it can allow water to move from this side going this side. At the same time, it is allowing water to move this side going this side. So it is permeable. It allows water molecules to diffuse through it. If a concentrated solution is separated from a dilute solution by a suitable membrane, water will pass from a dilute to a concentrated solution. So here we have water in a dilute solution, and this side we have water in a concentrated solution. Because on this side we have a dilute solution, it means that water is going to move from this side going to this side of the solution, which is concentrated. So we shall see that water is moving from the dilute solution and entering into the concentrated solution. But it does not mean that water cannot move from the concentrated solution going to the dilute solution. It does so. But when you look at it, you will see that the water which is moving from the concentrated solution back to the dilute solution is less compared to the water which is moving from the dilute solution going into the concentrated solution. In fact, water passes both ways, but it is faster from the dilute to the concentrated solution. We have seen that water moving from the concentrated to the dilute is less compared to the water which is moving from the dilute into the concentrated solution. You can see that we have a lot of water moving from the dilute to concentrated solution compared to the water moving from the concentrated to the dilute solution. So the water movement is a two-way flow, but it is faster from the dilute to the concentrated solution. There are microscopic pores in the membrane. This membrane that we have in the middle, it has very small pores which we are calling microscopic, and those very tiny pores are the ones that are allowing the flow of water in either direction. And because water is flowing from dilute faster than it is flowing from concentrated, that means that the level of the water in the dilute solution is going to go down, while the level of the water in the concentrated solution is going to rise. What we are seeing here, we can see that the level of the water in the dilute solution goes on decreasing, and that one in the concentrated solution goes on increasing. until when an equilibrium is reached. An equilibrium does not necessarily mean that the level of the water should be the same. An equilibrium here is whereby the concentration this side becomes the same as the concentration this side. So that's why you are seeing now the color of the water is the same, meaning that we have reached an equilibrium whereby all the concentration of the water is the same, but the levels are different. Molecules below a certain size can diffuse through the pores. We have said that we have pores in these membranes, and these pores, they are very microscopic holes which allow materials to flow across. Now, they only allow materials or molecules below a certain size to flow through those pores, meaning that the molecules which are bigger may not pass through those pores in the membrane. So those particles which are big, they will stay on one side, but the particles which are very small, they can move across the pores. Water molecules can easily diffuse through the pores. So we can see that the water molecules are easily flowing through the pores, moving to all directions. For example, if we take this structure here to represent a water molecule, that means that all these structures that we are having here, they are water molecules being represented by that structure. Those ones are water molecules. And if we take this structure to represent the sugar molecule, if that one is representing the sugar molecule, then this one here is a sugar molecule, and this one also is a sugar molecule, and this one is also a sugar molecule. There are many water molecules on the right as they are on the left, but many of them are attached to the sugar molecules, and therefore they are not free to move. So I want us to look at this one and we try to analyze. Where will the water molecules flow to? We have water molecules this side and we have water molecules this side. So let us try to see how many water molecules we have on one side. If we count the water molecules here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34. So on the left hand side, we have 34 water molecules. Let us now count this side and we see. have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35. They are 35. So if they are 35, that side, which means that water molecules will move in this direction. Why? Because here we have 35 water molecules, and this side we had 34 water molecules, which means that this side the water molecules are more than this side. So we expect the water molecules to flow in this direction. But is it going to be true? No. Why? Because now our water molecules, according to this diagram, it is going to flow in this direction. Why? Because in this direction here, we have water molecules that are held on the sugar molecule. Those water molecules held by the sugar molecules, they are not free to move, which means that they are no longer existing as water molecules. So that means that on this side now, the free water molecules are very less compared to this side. Therefore, water will move from this side and it goes to the other side until the free water molecules can balance on both sides. That is when we shall reach an equilibrium. Because they are more free moving water molecules on the left, more diffuse through the pores of the membrane from the left to right than from the right to left. So here we can see that more are going to diffuse from left to right compared to those ones that are diffusing from right to left. Why? Because on the left we have more free water molecules that can easily diffuse through the pores. This side, these water molecules that are held by the sugar molecule, they cannot go through here because they are held strongly by the sugar molecule. Therefore, they are no longer free water molecules. They cannot flow. So the only ones that can flow are these ones that are free. Those are the ones that are going to flow, and they are the ones that are able to flow through. But we need an equilibrium. Osmosis is going to be the movement of water molecules from a region of their high concentration, a region of high water molecule concentration, to a region of low water molecule concentration. So water is mainly going to move from left to right. Much of the water faster flow from left to right than from right to left. Sugar molecules can pass through the membrane, but being surrounded by a cloud of water molecules, they move more slowly. So these sugar molecules can go across. But the problem is that these sugar molecules are surrounded by water molecules. And because they are surrounded by water molecules, their movement is going to be very slow. So they will be moving very slow compared to these water molecules which are this side. Because the membrane allows only molecules of a certain size to diffuse through it, it is called selectively permeable. So here you can see that the small size will go through the pore. But if we have a bigger size, like this one of the sugar, it cannot go through here. Why? Because it is very big. So the membrane is just going to allow only molecules of a certain size to go through it than other molecules of bigger size. Therefore, that's why we are calling it selectively permeable. The cell membrane functions as a selectively permeable membrane. We have seen that selectively permeable membrane is that membrane that can allow some substances to pass through and other substances not to go through. Suppose if we are taking this one to be our cell, this cell, it will be having some pores through it, and those pores, they would allow materials of a particular size to go through them, and other materials will not be allowed to go through them. That is what we refer to as selectively permeable membrane, meaning that it can allow these materials to go through of the smaller size and also to come into the cell of smaller size. But if a material becomes too big of a particular size, then the pore is not going to allow this material to enter into the cell. Therefore, we call it now selectively permeable. It selects what should enter and what should not enter, depending on the size of the material. The cell sap and cytoplasm function as fairly concentrated solution. So we are looking here and saying that this one is our cell. If this one is our cell and this one has pores, which pores allow some materials to go through and others not to go through. So if this is our cell, we find that the cell and the cytoplasm and the cell sap are fairly concentrated solution compared to the surrounding solution around the cell. So if it is like that, that means that water molecules will flow into the cell faster than the water molecules flowing out of the cell. So that one would mean that if more water is entering the cell, it means that the cytoplasm of the cell and the cell sap are fairly concentrated solution compared to the outside world. Osmosis in animal cells. Let's look at osmosis in animal cells. There is a greater concentration of free water molecules outside the cell than inside. So when we look at our cell here, we can see that this is our cell membrane, which is surrounding the cell, and it is a selectively permeable membrane. Now, when you look at inside the cell, you will see that there are very few free water molecules inside compared to the outside of the cell. So that one means that water molecules will move from outside the cell and they enter into the cell faster than going out of the cell. So water diffuses into the cell by osmosis and the cell swells up. So as the water is entering, when the water is entering into the cell, it means that we are increasing on the amount of water which is entering the cell. And that water inside the cell now will exert a certain pressure on the cell membrane. As it exerts a certain pressure on the cell membrane, then the cell is going to swell as it is swelling outside because of water diffusing into the cell by osmosis. If osmosis continued, the animal cell would burst. So if we continue to allow in more water to enter into the cell, that means that we are creating a pressure against the wall to increase. And as this pressure against the wall or the cell membrane increases, it is going to cause the cell to burst. This would be bad news for animals. If the cell bursts open, then that is disaster to the animals. Because the animal, if the cells go on bursting, then the animal may not survive. Consequently, there are processes in the animal's body which control osmosis. There are some processes in the animal's body that control osmosis. Mainly, this is done by keeping the concentration of the body fluid outside the cell as the same as it's inside. So to prevent the process of bursting of the cell, we make sure that the concentration of the fluid outside the cell should be the same concentration of the fluid inside the cell. So that one will prevent the excessive flow of water into the cell, because the flow of water into the cell will be equal to the flow of water outside the cell. Therefore, the cell will not burst. Consequently, there are processes in the animal cell that control osmosis. Okay. In freshwater fish, this is freshwater fish, the blood is more concentrated than the surrounding water. So in a fish, you will find that the blood inside is more concentrated comparing it with the water outside. So there is more concentration in the blood than the water outside. As a result, fish take in water by osmosis. So because we have a higher concentration in the blood compared to the concentration outside in water. So what is going to happen is that the fish will take in water by osmosis. So water will enter the fish by osmosis. Not so much through the skin, which is thick, but through the gills, which have a very thin membrane. So much of the water is going to pass through the mouth, and then it will go to the gill. It will not much of it pass through the skin because the skin is very thick. The fish gets rid of excess water by means of its kidneys. The fish gets rid of its excess water by means of its kidney. So here we have the heart, which pumps blood, and when the blood goes to the gills, the gill absorb water by osmosis. When they absorb water by osmosis, water is going to be carried in the blood. So the blood carries water to the kidney. Now this blood, which is carrying water, it is going to transfer it to the kidney. And in the kidney, it's where we are going to expel the excess water, and it will come out in form of dilute urine. Seawater is more concentrated solution than the fish's blood. That will be osmotic. What will be the osmotic effect? Now here we have a seawater fish. The concentration of its blood is lower compared to the concentration of the water outside it. So what do you think will happen osmotically? Osmotically, if outside the concentration is high, then we are going to expect water to diffuse out of the fish, because inside the fish it has very low concentration of salts, and there is high concentration of salts outside. So water will diffuse from inside and it will go outside by osmosis. The seawater fish will lose water by osmosis through the gills. So we are going to see our fish losing a lot of water by osmosis through the gills. Why? Because there is less water outside of the fish and there is more water in the fish, or there is low concentration in the fish and high concentration of salts outside the fish. So water will flow through the gills going out of the gills or out of the body of the fish. The fish drinks seawater and the gills expel the excess salt from blood, so keeping its concentration constant. So we are looking at the fish, which is a seawater fish, what happens is that this fish is adapted in such a way that it drinks the seawater, and its gills, they expel the excess salt from the blood, so keeping its concentration constant. Because this is seawater, seawater is more concentrated. So because it is more concentrated, when it is taken into the mouth, the gills are going to expel water which contains more salts than the salt that entered into the mouth. So it is going to release a lot of salts outside so that it maintains a constant salt concentration inside the body. Seawater fish related to sharks, for example, dogfish, avoid losing water to their surroundings by maintaining their blood at the same concentration as the seawater. So for them not to lose their water, for the seawater fish like the dogfish and so on, what they are going to do, they are going to maintain their blood at the same concentration as that of the seawater, so that they don't lose water to their surroundings. We know that the seawater contains a lot of salts. That would mean that the fish would lose water to the seawater because of the high concentration of salts in the seawater compared to that in the body of the fish. So what the fish does is to maintain the same concentration as that one outside. So inside the fish, the concentration is going to be the same as that one outside in the seawater. So the net flow of water will be the same. Water coming in will be the same as water going outside because of the equilibrium. In plant cells, the cell membrane acts as selectively permeable membrane. That is in plant cells. The cell wall is freely permeable to water. The vacuole contains solutions of salts and sugars. So we have the plant cell. It has a cell wall outside and a cell membrane inside. But the cell membrane is selectively permeable, whereas the cell wall is freely permeable to water. So the vacuole, this one contains salts and sugars, And because it contains salts and sugar, that means that the concentration inside is very high. And because there is a high concentration of salts inside the vacuole, that one is going to attract water to enter into the cell from the surrounding. Because the cell wall is freely permeable to water, but the cell membrane is selectively permeable. So because there is a high concentration of salts in the vacuole compared to the outside, water is going to flow through the cell wall and the cell membrane into the vacuole. If there is water outside the cell, it will diffuse by osmosis into the vacuole. As we have said, inside the vacuole, we have a lot of salts and sugar. So that makes the concentration in the vacuole being high compared to the concentration outside the cell. So the concentration outside the cell is low and the concentration inside the cell is high. This is going to force water to enter into the cell vacuole by osmosis, moving from a region of high water concentration to a region of low water concentration, or moving from a region of low concentration to a region of high concentration of salts. The vacuole will expand, pushing the cytoplasm outwards against the cell wall. Now, because the water is flowing inside, now the vacuole is going to expand. And as it is expanding, it is going to apply pressure onto the cell wall. And this is going to cause the cell to start swelling. The cellulose cell wall cannot stretch, so the vacuole cannot continue to expand. So as the vacuole is expanding and applying pressure to the cell wall, time reaches when the vacuole can no longer expand anymore, because the cellulose cell wall cannot be stretched any further. So more pressure applied by the vacuole will stop, because the cellulose cell wall is not stretchable. But the pressure of the vacuole against the cell wall makes the cell very firm. So because of this pressure, which is applied by the cell vacuole, it is going to make the cell to become firm. The cell becomes firm because of that pressure applied by the vacuole against the cell wall. Plant cell in this condition is said to be tagged. It is tagged because you can see that it is receiving a lot of pressure from the cell vacuum, and that one makes it to become very firm, or what we call being tagged. If all cells in the plant are tagged, the plant will be firm and upright, and the leaves will be expanded. So when there is a lot of tuggidity in the plant, that means that the plant is firm and it will stand upright. Its leaves will be expanded. Osmosis between cells. Let's look at osmosis between cells. If the concentration of the cell sap is greater in one cell than its neighbor, then water will pass by osmosis from the less concentrated to a more concentrated cell. Now let us look at these two cells. We have the first cell here, and this first cell, we are saying it has got a sap that is more concentrated. Then when you look at the second cell here has got a cell sap which is less concentrated. Therefore, water is going to move from a cell sap less concentrated to a cell sap which is more concentrated. So water will flow from this cell and it will enter that cell. Osmosis between one cell and the next plays the part of the movement of water throughout the tissue in the plant. So this osmosis which is taking between these two cells is playing a part of water movement from one part of the plant to another part of the plant. So water is now flowing in that direction because this one has more water and this one has less water. Therefore, water will move from where there is more water where there is less water. And that one will continue to another cell like that, and then water will be moving throughout the tissue in the plant. These cells are short of water and the tissue is limp and the plant is wilting. So when you look at these cells, you can see them. They are becoming limp, meaning that they have less water. If they had a lot of water, they will be tagged. But because they have lost water, therefore the tissues are becoming limp. And if they become limp, then we shall say the plant is wilting. If you look here, you can see that our plant here is wilting. The leaves are curling. These curling leaves, it is a sign of loss of water by osmosis. The leaf cells are no longer tagged and the leaves curl up and droop. Because the cells are losing a lot of water and they are becoming limp, so they are no longer tagged. They are not receiving a lot of pressure from the cell vacuum. So they lose their tuggidity. And the leaves, what is going to happen to the leaves? The leaves are going to curl up. They will fold and they will droop, meaning that they have lost a lot of the water by osmosis and the cells are no longer tagged. The cells have taken up water by osmosis. The cells are tagged and the tissue is firm. Now, if we look at these cells, we can see that these cells have taken in water and they are tagged. Tuggidity means that they are swelling. If you look at them, you can see that they are swelling, meaning that they have taken in a lot of water. And when that one happens, then the cells are going to become tagged and you will see the leaves being straight and flat because they are tagged. So the tissue, when you look at the tissues here, the leaves are very firm. Water is one more available. Water is once more available. The leaf cells, they will take in up water by osmosis and they will become tagged. So when water is available, the cells will take in water, and when they take in water, they will become tagged. And when they become tagged, therefore our plant is going to be very firm, as you are seeing it here, because it has got more water than the environment. The leaves are now firm and they are held horizontally. If you look at our leaves now, the leaves are firm and they are held horizontally. Woody plants, such as trees, shrubs, these ones, they do not collapse when they wilt, but their leaves will limp. For the woody plants, when they lose water, they do not collapse, but they wilt by their leaves becoming limp. So we can know that it is wilting by looking at the leaves, which are becoming limp, but the stem will remain firm. In the growing region of the plant, the cell walls are not inextensible, but are still plastic. So we have here our plants which are growing. These are shoots, and you can see that on the tip of the shoots we have cells which will continue to divide. Here, these are cells which are dividing, and after division the vacuoles will start forming in those ones which have stopped dividing. Then later on, those cells now will absorb water by osmosis and they are going to expand. As they are expanding, then we say the plant is growing. Cell division takes place in these cells at the tip, whose cell wall is inextensible, but they are still plastic. So when we come to this region, this is a region where we are having the cell absorbing water and it is expanding, and the virtue is the one taking in water, continues to expand. And there we say the plant is growing. When the virtue takes in water by osmosis, it expands, pushing out the plastic cell wall and increasing in the size of the cell. So you can see here we have the virtue, and that virtue is absorbing water. And when it absorbs water, it is going to push on the plastic cell wall. As it pushes on the plastic cell wall, there is going to be an increase in the size of the cell. The cell wall at the growing point increases the number of cells, but the cell expansion causes the increase in size. So when you look here, we have regions. This upper region here, it's where cells are dividing. And when we come to these regions here, it's where the cells are expanding. So when we say cell walls at the growing point increase the number of cells, here we are increasing the number of cells where we have cell division. But cell expansion is taking place here where the virtue absorbs water by osmosis and it expands. As the cell receives pressure from the cell virtue, the cell increases in size. So it is cell expansion which causes the increase in size. But at the tip, we are just increasing in number. When you come down at the cell expansion, we are increasing in size. Cell division followed by cell expansion causes growth. At the tip, we have cells that are dividing, and when they divide, they increase in number, then followed by cells which are expanding. And as the cells continue to expand, then that whole process is what we will cause growth. The osmotic force generated by the cells and tissues are very large. This is why you get weeds pushing through the tarmac. So there is that osmotic pressure, that osmotic force, which pushes the water into the plant or into the vacuole, and then the vacuole will apply pressure. The vacuole will apply pressure on the cell wall, which will become tagged, and because of that tuggidity, the plant becomes strong. And this is what is going to cause the plant to push out of the tarmac. It will come and push out of the tarmac because of the osmotic pressure exerted by the vacuole onto the walls of the cell wall. The cell here has just divided. It was one cell, and then it has divided to form two daughter cells. The vacuole starts to form in one cell. So the cell which has divided to form two daughter cells, one of the cells is going now to start developing vacuoles. It will develop vacuoles. Vacuoles are filled with water. The vacuoles, they absorb water by osmosis and stand to extend the cells. So when these vacuoles, which have been formed, they absorb water by osmosis, they are going to start extending the cell. So the cell will be growing and being pushed by the pressure exerted by the vacuole after absorption of water by osmosis. The vacuole begin to join up and continue to absorb water and extend the cell. So when you look at the pieces of vacuoles, they go on filling up and then joining together. And when they do that, they exert a pressure onto the cell wall, and this pressure is the one that is going to cause the cell to extend. Note that the cell expands in length rather than its width. So when you look at the cell is increasing in size, in length, is increasing in size, in length, not in width, not in its width. It's not increasing in size in width, but it is increasing in size in its length. So when you look at this cell and this cell, they have the same width, but they have different length. Different length. This is due to the arrangement of the cellulose fibers in the cell wall. What is causing this type of growth is the arrangement of the cellulose fibers in the cell wall. Thank you very much for participating in Osmosis. Please do like, share, and subscribe.